Welcome everyone. Myself Shilpi Banerjee will be discussing about library screening. Well, we all very well know what is library, right? Library is collection of something. So, as we are here to discuss about genetic library, so genetic library screening will be something which is identification of DNA or identification of clones or something like that, right students? So, let us now explicitly see the definition of what is library screening. Library screening is nothing but the process of identification of the clones carrying the gene of interest. Screening relies on a unique property of a clone in a library. The DNA libraries consist of a collection of probably millions of clones in the form of either plaques or colonies on a plate. Screening of libraries can be done by following approaches based on number 1, detection of particular DNA sequence, and number 2, expression of a gene. Screening a genetic library is a process that involves identifying specific DNA sequence or gene of interest from a huge collection of genetic material. Genetic libraries are created by fragmenting an organism's DNA and inserting them into vectors which are vehicles or a medium that allows for the replication and expression of the DNA fragments. Let us now see how basically a screening of a genetic library is typically conducted. There are several steps. The first step is creation of a library. So, this step involves isolating the DNA from the organism of interest and fragmenting them into manageable pieces. These fragments which have been isolated are inserted into vectors which can be either plasmid or some other vehicles used for DNA replication. Now, this results in collection of vectors each having different DNA fragment. The second step involves choosing a screening method and screening procedure. Now, depending on the goals of research, different methods can be used to screen the genetic library, such as functional complementation. This method is used to identify genes that complement a specific phenotype of function in a recipient organism. Next is hybridization. So, let us say if we have a probe, a labeled DNA or a RNA molecule that matches with the gene of interest, we can use it to screen the library. The probe DNA or RNA which hybridizes with its complementary sequences in the library allow us to identify vectors carrying the gene we are interested in. Next is polymerase chain reaction screening. So, if we have some information about the gene's sequence, we can then design primers that are specific to the gene. By using these primers in a polymerase chain reaction, that is PCR, we can amplify the gene of interest from the genetic library. The third step is, after all the screening method and its procedure, isolation and characterization. After we have identified the vectors that are carrying the gene of interest, these vectors are isolated and characterized. Now, this might involve sequencing the DNA to confirm its identity or conduct further experiments to study the function of gene. Next step is validation and further studies. Once the gene is identified, its role and function can be further investigated. This might involve gene expression studies, characterization of protein, or studying its effect in different biological contexts. Next is iterative process. In case initial screening does not give the desired results, researchers may modify their screening approach, probe, or other methods involved. And this iterative process can be repeated if required. Now, let us see some of the techniques which are used for screening of libraries. First is hybridization probing. This is the screening of a library with labeled probe, let us say radioactive, bioluminescent, etc., to identify a specific sequence of DNA or RNA. This can be done either using colony hybridization probing, wherein we search for a specific DNA sequence which is mixed in a population of transformed bacterial host cells, or by using plaque hybridization method, wherein we screen bacteriophage plaques. Next technique is chromosome walking. So, chromosome walking is nothing but where overlapping fragments of a particular chromosome is utilized and it is isolated for gene of interest which may be present upstream and downstream from the original DNA fragment. Next step is polymerase chain screening. The primers which are designed to match the sequences of interest are used in a polymerase chain reaction to amplify the targeted DNA from the library. The next method is fluorescence in situ hybridization. It is basically a cytogenetic technique which is used to detect and localize the presence or absence of specific DNA sequences on chromosome. This technique can also be successfully used to detect our gene of interest within the mixture of various recombinant clones. Next method is molecular beacon. Now, molecular beacon is a fluorescent probe molecule that is basically designed to produce fluorescence color only when it binds with a specific DNA target sequence. Next technique is gene tagging. This method is applicable only when we do not have any information regarding the base sequence of our gene of interest, but yes, it can detect a specific phenotype encoded by it. This method relies on integration of known sequence of DNA, preferably at random sites in the genome of the host cell. Next method is yeast-2 hybrid system. So, there are two hybrid proteins, one of them is fused to a DNA binding domain and the other one is fused to an activation domain which are expressed in yeast. So, interaction between these proteins lead to the activation of a reporter gene which is then investigated. Next technique is immunological screening. So, this method aims at identifying the protein product of a cloned gene. The technique requires that the protein is expressed in recombinant. So, in this method, instead of a nucleic acid probe, a specific antibody is used. Next technique is fluorescent activated cell sorter. So, fluorescent activated cell sorter is a machine that can rapidly separate the cells from a mixed suspension of mixture of cells on the basis of presence or absence of fluorescence. This method is used to screen those genes of interest whose protein products are expressed on the surface of a cell. Next is Southwestern screening. This is used when the gene of interest encodes for a sequence specific DNA binding protein, whereas the next technique, which is Northwestern blotting, is used when the gene of interest encodes for a sequence specific RNA binding protein. So, the major difference between southern screening and northwestern screening is Southwestern screening encodes for DNA binding protein, whereas northwestern blotting encodes for a RNA binding protein. Next technique is functional complementation. A host organism which lacks a certain function is transformed with a genetic library. If a gene in the library complements the missing function, the host organism will regain the function. Next is screening based on in vitro translation of mRNA, which is basically categorized into following two types, that is HART and HRT method. Now, what is HART? Hybrid arrest translation. HRT refers to hybrid release translation. Now, let us see the details. The HART method is a technique which is used in the molecular biology to identify and isolate the genes of interest based on their functional complementation of a specific phenotype. This approach is particularly useful for studying genes that might be difficult to screen using traditional methods. In hybrid release translation, that is in HRT method, a colony hybridization of cDNA library is carried out. The mRNA isolated from the cells are added to the nitrocellulase paper having the single stranded specific cDNA. Now, the mRNA of interest will bind to the cDNA, whereas the rest of the mRNA will not be hybridized. Finally, the unbound mRNAs are removed and the hybridized mRNAs are isolated by denaturing the cDNA mRNA hybrid. These mRNAs are added to the cell free translation machinery where proteins are synthesized. The size of the protein can be further analyzed by method of SDS page. Library screening has wide range of applications across various scientific fields. This technique is systematically used through collection of items to identify specific elements that possess desired properties. Let us now discuss about some of the key applications of library screening. Number 1, drug discovery and development. By library screening, it is quite easier to identify potential drug compounds with specific therapeutic effects. Next, genetics and genomics. For identification of genes associated with specific trait, function or disease, genetic library is quite beneficial. Next, for interaction between protein. To understand the cellular pathways and protein networks by identifying the interaction between proteins, genetic library is very much helpful. Next application includes phage display and antibody engineering. Generation of novel peptides or antibodies with specific binding properties is because of library screening. Next, for identification of genes essential for specific cellular process via functional genomics and RNA interference, genetic library is beneficial. Next application, for identification of enzymes which are used as biocatalyst and in enzyme engineering. So, basically to develop enzymes with improved catalytic properties for different industrial application, genetic library is beneficial. For optimizing microbial pathways in order to produce biofuel, chemical or pharmaceutical, or in case of metabolic engineering, gene library plays a very important role. Next application includes studying evolution of protein and their function. Okay, let us now discuss about library screening in next generation sequencing, which involves preparing and sequencing DNA or RNA samples to identify specific sequences or variants of interest. It is a crucial step in genomics research, drug discovery and various biological studies. Researchers create libraries by fragmenting DNA or RNA, adding adapters and amplifying target regions before sequencing. Then through bioinformatic analysis, they identify and interpret the sequences to understand genetic information or variations within the sample. Now, let us see method of library screening in next generation sequencing. First step is preparation of sample. DNA or RNA is extracted from the sample. It is then fragmented into smaller pieces. These fragments are often tagged with adapters that allow them to be attached to sequencing platforms. Second step is construction of library. Fragments are ligated to adapters which serve as primer binding sites for sequencing. This step can also include PCR amplification to enrich the regions of interest. Next step is hybridization or target enrichment. In some cases, hybridization probes or PCR primers are used to capture specific regions of interest from the library, allowing for targeted sequencing. Now, this is useful for focusing on specific genes or genomic regions. Next is sequencing. The library is now loaded onto a NGS platform where high throughput sequencing generates millions of short DNA or RNA reads. Various sequencing technologies like Illumina, Ion Torrent and PacBio are used. Analysis of data. The raw sequencing data is processed and aligned to a reference genome. This step involves quality control, removal of adapters and mapping which reads to their genomic position. Next is calling of variant. If our goal is to identify genetic variation, the mapped reads are compared to the reference genome to detect single nucleotide variants, insertion, deletion and other structural variants. This step is followed by annotation and interpretation. Variants are annotated with information about their potential effects on genes and proteins. This helps researchers to understand the functional significance of these variations. Next is visualization of data. Results are often visualized using tools that display the location and type of identified variants, helping researchers to draw meaningful conclusion from the data. Overall, library screening in NGS allows researchers to efficiently explore genetic information, discover mutation and analyze specific genomic region with high precision. Now, let us see the process of library screening by using NGS method. Now, let us see about library screening in future. What is the role of library screening in future generation? In the future, library screening in next generation sequencing is likely to see advancement in several areas. First is higher throughput and speed. NGS platforms are likely to become even faster and capable of processing larger volumes of sample simultaneously, which will reduce the time required for preparation of library and sequencing. Next is it will improve the accuracy and sequencing of long reads. Technologies that offer higher accuracy and longer read lengths will enhance the ability to accurately detect variation in complex genomic region. Next is single cell sequencing. Library screening at the single cell level will become more common, which will allow researchers to explore genetic diversity within individual cells and understand cellular heterogeneity. Next is integration of multi-omics. Integrating NGS data with other omics data such as proteomic, metabiomic, epigenomic will provide a more comprehensive understanding of biological system. In the future, artificial intelligence and machine learning will also be beneficialized by using library screening. Advanced AI and machine learning algorithms will play a significant role in data analysis, helping to identify pattern, predict functional impact of variants and enhance the interpretation of complex sequencing data. So, over the past decade, there has been an exponential increase in the development of new library screening method, which is very much essential for efficiency, for result oriented, for time oriented results. The efficiency of PCR based technique has been increased with the introduction of new primer design tactics, pooling schemes and improved thermal cyclers. Hybridization based approaches, which includes colony plaque hybridization and bacteriophage hybridization, they have been streamlined due to the creation and modification of labor saving devices such as robotic plaque or colony pickers and gridders. Though a working draft of the human genome is now complete, other large scale sequencing projects are continuing to provide the drive to further advance screening techniques. So, as we have seen that library screening is very much important to know about all the data which can be applicable right from drug designing to agricultural application for detecting new genetic disorders and anything which is very much closely associated with human body. It becomes very important to identify, to create, to analyze all the genetic components present in the huge genetic library. New developments are going on, but by using the traditional methods we can explore new possibilities about how labor saving, time saving and more efficient methods can be created which can help to analyze gene from the huge collection of genetic data. Thank you everyone. I hope this was a brief insight about how library screening is applicable in genetic engineering.